Okay, all right. And my heart stopped pounding. All right, we're going to now get into culture and identification of infectious agents. Okay, again, we are going to go through these keywords. Now, this one's a little different. In this case, what I've done is we're talking about the major issues that we're going to be discussing in this lecture, not just terms. Because this is a little more mechanistic than the previous lecture, which was the introductory lecture. First of all, we're going to talk about about growing and isolating bacteria. How do we organize bacteria taxonomically? Taxonomy is extremely boring, and it's basically a way of organizing your closets, is a good way of looking at it. But we have to know a taxonomy because it's a language. It's just like any other language that you learn in different jobs that you have. You have to learn a medical language. Well, if you can work with bacteria and infectious diseases, you have to know the language. So we're not going to focus so much from a point of view of taxonomy, but in terms of really making some sense of the myriad of awful Latin terms that we're going to learn over the next couple of weeks. And that's what that's all about. Now, we're also going to talk about what's done most of the time in the clinical laboratory, which is after culture. Basically, we grow bacteria up in this fashion, and then we deal with them. And what I'm going to try and get across is what's routine, what's state-of-the-art, what do you need to know, etc., etc. And we're going to talk a little bit about direct detection without culture, which is predominantly the PCR or polymerase chain reaction. Okay, so that's the idea. This will give you the overview of what we're going to be doing and why we're doing it in this order. Okay, to use a different term than organizing your clothes in a closet, taxonomy defines common traits among strains for a bacterial species. What on earth does that mean? The point is that you're all strains in this room. Some of you have got red hair. Some of you have got dark hair. Some of you are small. Some of you are big. But you're all human beings. You're all members of the same species. You are strains. Okay? That's what a strain is. An individual example, and it's the most common term that we use in bacteriology, along with species. Species is certainly the most common. And after species, perhaps strain. If you don't know what a species is and you don't know what a strain is, no offense, you don't know anything. Okay? You have to know what a species is, and we'll get more into this as we go along. You have to know what species and strains are. Now, the other taxonomic terms come up now and again, and I'm keeping them to the minimum in this course because we don't use these terms. When we'll use them in the individual lectures, and I'll give an example in a little bit. In this day and age, taxonomy is usually genetic, or in other words, genomic. I was surprised to discover that you can actually pay a company for $10,000 that will actually give you all the bits and pieces that could make up an entire genome for $10,000. It used to take, 2 or 3 years ago, it took about 30, 40 people and tens of millions of dollars maybe to get that information. Now, that doesn't mean for $10,000 what you get is a genomic sequence. What it means is you get bits and pieces of the genomic sequence and you have the job of putting it together. But that's the situation we're in at the moment. We're in a genetic age, we're in a genomic age in terms of taxonomy. And I didn't say in terms of the clinical microbiology laboratory. And that's a big difference you'll see as we're going along. The taxonomy is usually genetic, and in this day and age it's even becoming genomic. This basic information, however, is the fundamental information that companies need to make kits. If you don't know anything about the genetic makeup of an organism, you can't make a kit. It's as simple as that. The taxonomy is the basic research that's done to give you the information that's needed so that you can come up with diagnostic kits. Okay, let's talk a little bit more about species and strains. I think you've probably got it already, but that was a new one today. This one I've been using for a few years in different places. Let's imagine that you were a Martian and you came down here to Earth and you knew nothing about the life forms on this planet. And you saw these strange animals roaming around. Maybe it was a circus. I don't know. Who knows? Maybe the animals were sort of these weird colours. But anyway, there was brown pigs and black cows and brown cows and multicolored cows and what have you. And there were these strange pigs roaming around as well. The Martian looks at these two animals and says, gee, well, on Mars, everybody's brown. These must be the same species. Makes sense, doesn't it? They're the same species, right? Cows and pigs? Boy, you guys need to go to sleep. Come on. Of course they're not. They're not the same species. We all know that. What the trait that is determined, the fact that they are the different species, is the fact that it's not colour, it's morphology, shape in this instance, of course other features. So the bottom line is that just because, the bottom line is you're always going to see variability of traits among individual members of a species. You're always going to see this. But if you pick the wrong trait, in this instance, colour, you have no information. Zero information. Colour here in no way would tell you that was a cow and that was a pig. Okay, and that's the whole reason why you have to do a lot of work to define the molecular characteristics of particular species. You want in a test, you want something that is conserved among all the members of that species that you're interested in, but not among the others. And in this simple case, it would be the shape or morphology. Now, there are various categories in taxonomy, and those of you who've had some zoology and botany in the past are well familiar with these terms. And you may even have zoologists and botanists who love this stuff. Well, I don't. Sorry to inform you that. I've done a lot of taxonomy, but I don't love these terms. The bottom line is that it's going from the smallest to the largest grouping. That would be the individual member of a species, a single isolate or line. The type would be a group of strains. Very vague terms, purposely so. A species would be a group of genetically related strains. I'm not going to give you any definitions because they've changed a lot in the last few years, and who cares? But the species would be a group of related strains genetically. The genus would group together species that have some relationships genetically, and the family would include multiple genera. The most common terms that we use, and that's why I put it in red, is the term species. But we must know about the word strain because there are many diseases that we're going to discuss which are caused not by certain species, but are caused by certain strains or types within that species, which have particular pathogenesis characteristics. And so if you come away with the idea, one species, one disease, well, you'll fail the second part of the course. Okay? So that's an important point to understand, that diseases can be at the species level, or they can be strains or types within the species level. We very rarely use the word family. Perhaps the exception being with the Enterobacteriaceae, and I will use that term in my lecture on the enterics. But generally speaking, these terms, the term family is very rarely used. To give you an example, I don't know if let's see if I've got another slide here that has this, I don't remember. Probably not. But in the handout, there is a good example. I want to just briefly cover it. Species. Okay. Let's take the species Streptococcus pyogenes and the species Streptococcus agalacti. Again, you don't need to take notes on this. This is the definitions in the handout. Just make sure you understand. Okay, in the case of Streptococcus pyogenes, the species name would be Streptococcus. The pyogenes part of the word, the genus part of the word would be Streptococcus. So Streptococcus pyogenes, the first word tells you what genus it's in. The second part of the word is the species word. So Streptococcus pyogenes would be, and Streptococcus agalacti would be two species within the genus Streptococcus. Okay? So that's how we use these words, species and genus. So even when we're talking about a species, even if we don't say it, we're always using species and genus terms. Escherichia coli, again, it's the genus Escherichia. Salmonella, the genus Salmonella. There can be multiple species within a genus. Okay, so that's how we use these terms. And there is an example given in the handout, as I said. Okay, so that's classification. Now, identification of infectious diseases in a diagnostic laboratory is an applied area. It is not basic research. It is an applied area designed for a particular purpose to give you information about your patient, and preferably as quickly and accurately as possible. So it uses taxonomic principles, but it certainly doesn't use, there may be people on the side, of their own interest doing some research, but its clinical lab does not develop kits. It doesn't do molecular taxonomy. It uses these things. It uses these principles on an everyday basis. Because first of all, knowing what you've got aids treatment. Knowing that you've got Mycobacterium tuberculosis, which takes several weeks to grow up in the laboratory and is a very slow infection that takes months to develop, is a very different situation with having a meningitis caused by Streptococcus pneumoniae where you'll no doubt be dead in a day or two. Okay, so it is useful to know what the organism you've got, where is it present in the body, and what is it? And that's what the clinical lab does. So it aids treatment, first of all. Secondly, in some instances, it helps antibiotic selection. Because you can do simple tests, and I'll give you a couple of examples and we go on. You can do simple tests that can tell you whether an organism is killed by a particular antibiotic or not. That helps you select the antibiotics. Now, I do not want anybody to come away from this lecture with the impression that basically what you do as a physician is you sit there and you wait till the clinical laboratory gives you a result and then you start treatment. Because, I don't think your mentors will ever allow this to happen, but the result would be very simple. You'd have a lot of dead people. It takes a day or two to get results back, usually in the clinical laboratory, just to grow them up and tell you what you've got. The antibiotic susceptibility tests take even longer. In many cases, you'll only use them in a situation where you're having a problem with a patient. They're not responding to a particular antibiotic, and you might want to come up with another one. You don't sit back and wait. What you do is you treat with the best knowledge that you have, and you get the results back, and hopefully it will help you. And certainly does in some instances. Now, reference laboratories can be large clinical laboratories in big hospitals, big medical schools. We don't have a reference clinical laboratory here. And basically, the places like the CDC, and what you're doing is more sophisticated tests when you've got some unusual things going on. And in recent years, we're all aware of the unusual things going on after the events of 2001 with the Twin Towers and the subsequent anthrax attacks in 2002 on the country. So, that's where the reference lab comes in, to doing things that the regular laboratory just can't handle. And sometimes the reference laboratories, such as the CDC, will develop new tests. In this day and age, I would still say that the culture is still the backbone of the clinical microbiology laboratory. So let's talk about what we do in a regular clinical microbiology laboratory. The first thing you're going to do is you may grow the bugs on some liquid first and what have you, but at some point you're going to have these bacteria growing on culture plates. The reason you want them growing on culture plates is because bacterial identification is based on the fact that you have a pure culture. You cannot usually do, it's very difficult to do bacterial identification in mixed cultures because one organism may respond to one test, the other one might respond to another test, and then you've got, well, which one is it? It's both or neither. So generally speaking, you want a pure culture. So you may grow the organism up first, in some instances, in liquid broth to get large amounts of it. But it always ends up on a culture plate, or usually ends up on a culture plate. On a culture plate, you can look at the size, the texture, the colour, and whether, if you grow it on a sheep blood agar plate, whether the blood is lysed or not. And that's very important in some bacterial identification purposes, such as streptococci. You can also see whether they use oxygen or not. And we have a whole lecture on metabolism, and you'll realise at that point that these are bacteria, not human beings, and there can be all sorts of things going on in bacteria as regards metabolism, which doesn't happen as regards human beings. And one of the issues is some bacteria grow totally aerobically. They're killed by the absence of oxygen. And then there are other bacteria that actually have the opposite effect. They actually will only grow in the presence of oxygen. So anyway, whether they grow or not in oxygen, aerobic versus anaerobic, these are sort of a couple of the issues that relate to identifying bacteria. Let's take an example. This is actually taken from the CDC. It's a nice, simple demonstration. If you look at Bacillus cereus, for example, you'll see that this blood has totally been lysed. You'll see the individual colonies on the right-hand side, though, of the bacteria. Here you can't even see the colonies because they're just they're there, but you can't, there's nothing to, this green colours come from the lysed blood. So basically, this is referred to as hemolytic, and that's referred to as non-hemolytic. And I'll give you some other examples of that later on. So you can tell that one of the ways you can tell the difference between Bacillus anthracis and Bacillus cereus, for example, is by hemolysis. But that's not the most important thing in this course. I'm just giving it as an example of hemolysis reactions. I talked about mixed colonies already. By definition, you could have a situation, for example, if you isolated something from the skin, you're always going to get a mixed colony. The skin is covered in many, many different sorts of bacterial species. And you're always going to get different size colonies, different color colonies, and what have you. Most bacterial colonies are going to be opaque or white in color. Staphylococcus epidermidis, one of the major things isolated from skin, for example, produces yellow colonies, but most bacteria don't. But the point is now you can say, aha, well, I'm not interested in those yellow ones in this instance. I want to look at these white ones. So I can take that white one and remove it from the plate and grow it in pure culture. If you don't do that, if you don't separate first by the separation of the individual colony types on the plate, you're going to have a mess. Okay? Because you don't know whether you test, are you testing for that or are you testing for that? Who knows what you're testing for? So that's why we grow things on plates. Okay, the second thing that we do is we do a gram stain. The cells are observed under the microscope. And I said that I gave you a little bit of detail, gram-positives versus gram-negatives last lecture. Now we're going to get into the little more detail. Okay, you need to understand the mechanisms of the gram stain. You need to understand every step of the gram stain, and you need to understand what happens if you leave out the individual reagents. If you do anything different, you need to know, you need to think about it, understand what's going on, and make sure that you can make a prediction. I will go through this and explain what's going on and give you a couple of examples. All right, let's take a gram-negative organism. Now, we've said they're not usually coloured. Okay, if you look under the microscope. The colonies might be coloured, but when you look under a microscope, you usually don't see bacteria. Really, you can hardly see them at all, if you can see them at all. Now, what happens when you stain with the dye crystal violet, it has a word violet, but sometimes they can look blue when they're stained. It depends on the organism. Sometimes they stain more intensely or less intensely, and that's why it doesn't make a blind bit of difference if I say blue, purple, or any variation thereof, or anybody else does. I say that because in the past, people have drove me crazy, but you said it was purple next year. Well, you said it was blue. It doesn't make any difference. Purple, blue, any variation thereof. And the same with these, they will be blue at this point, or purple, or whatever. Then the iodine actually fixes the dye in place. It fixes the crystal violet. Without the iodine, the crystal violet would have a hard job of staying there in some places. The next step that's done is, there are different versions of this, but they usually involve alcohol or some other organic solvent such as acetone. The key issue, there's a destain step here. Now, in the case of a gram-negative organism, it loses its staining much more readily than a gram-positive organism, and you can relate that to the structures of the cell envelope, and it's beyond the scope of this course for me to get into that. If anybody cares to, we can talk about it sometime, but not in class. Okay, now the issue comes down to is that the alcohol decolorizes the gram-negatives, and it doesn't decolorize the gram-positives. Now the final step is we use safranin, which is a pink or pinkish stain. And what happens is the gram-negatives now become pink because they were decolorized. This crystal violet stain is so intense that you don't see the pinkness there. It doesn't show up. So the color of the gram-positive doesn't really change when you treat with safranin. Bottom line, you end up with a pink or blue organism. But what you need to think about, and I'm not going to go through every permanent combination here, you can think about what would happen, for example, if you left out the crystal violet. Well, if you left out the crystal violet, then clearly there's no way, nothing would stain blue. The only thing that could happen was everything would stain pink. I'll let you think about that one later, okay? So that's the way, I want you to think about what happens with these different steps, okay? What would happen, okay? All right. That's the gram stain. It's described in detail in the handout as well, and you've got this slide, so take a look at it. Think about it. Okay, gram stain morphology. Now, you get more out of a gram stain than simply the issue of is it pink or is it blue? Is it gram-positive or is it gram-negative? There are other issues that come into play, particularly shape. There aren't many terms and combinations, really, of the shapes that bacteria can have. They basically only fall into a few types, being sort of round. Now, you'll notice this is what I was talking about just now. That is not referring to the genus Bacillus, right? Because it's not italicized and it's not got a capital B. Okay, but anyway, the issue is, regardless, you can make, when you read about this stuff, look at the way the words are being used, okay? Here we're just talking about, this is a generic term here, bacillus, and that's where it's been used as a rod-shaped as opposed to a round-shaped organism, a coccus, okay? So bacillus would be a rod-shaped organism. And they can be spiral or curved, an example of the spirochetes. And we will show you some examples as we go along. We'll show you some pictures. There's a nice website where they've got there's nice pictures of all the different bacteria under the sun. And we've picked some of the more useful ones to show them in the individual lectures later on. But anyway, they can be spiral or curved. And the other issue is they can be single cells, or they can actually, during the process of growing and dividing, sometimes they don't fall apart. They actually stick together. They can form a clump, or they can form a chain of cells. So, and that's the difference between staphylococci and streptococci. You'll notice here I didn't say staphylococcus and I didn't say staphylococcus, and you'll notice again it's not capitalized. When you use a plural like that, you usually, and I may make some mistakes, Dr. Mayo may make some mistakes in the quizzes and the exams and what have you. It won't affect the answers because you'll know what's going on. But the bottom line is when you do this, if it's plural, like cocci as opposed to coccus, it's not capitalized. So again, genus or species names would be capitalized or italicized or underlined. They can be underlined rather than italicized. And if they're plurals, they like that, like cocci being the plural of coccus, that would be like that. Again, gram-positive or gram-negative. Take an example. You don't have to be a surgeon here or an ophthalmologist or something, to know that that's pink and it's long. That's a gram-negative rod. Okay? That would be a chain by definition, because we can see the individual cells are these round cocci, the singular being coccus. That would be a streptococcus by definition, because it's occurring in a chain of circles or spheres. That doesn't mean it's always going to be a streptococcus. If all we had to do was look under a microscope and say, oh yeah, there's a chain there, it's a streptococcus, why would we need a clinical microbiology lab? I mean, it would be so simple, it wouldn't be worth even messing with it. But it's not. What these are, these tell you it could be a streptococcus, or it's probably a streptococcus. It doesn't tell you what species. It doesn't tell you, for example, if it's Streptococcus pyogenes, the causative agent of strep throat. It doesn't tell you it's Streptococcus agalacti, the major causative agent of neonatal septicemia and meningitis. You need more information than that. But it's the first place to begin. You get information on the shape of the cells, whether they're rods, spheres, spherical, spiral-shaped. They can be pleomorphic, which means variable in shape. There are a number of different possibilities. That's the place to begin. It's not the end, it's the beginning. So that was step 2. We first of all isolated a pure colony. We secondly looked at it under a microscope. The third thing is now we're really getting into the tests. We speciate. The standard tests for many, many years, some of them going back 100 years, are physiological tests based on basically does a bacterium produce a particular enzyme? Does it utilize certain sugars or amino acids or fatty acids or what have you? And these tests really, many of them have not changed over the years. And they've been with us many, many years. They're very simple and they work extremely well. We're in a transition point at the moment. We haven't got there yet between physiological, and I mentioned already, these molecular diagnostic tests, because we have simple, cheap tests here that are easily learned, and we have more complicated tests that require a bit more training. And it's really an issue of, what do you want to do that works the cheapest and the best? And most of the time, these physiological tests are just very easy to do, so they stay with us. But molecular tests are being used in most of the major medical centers around the country in addition to physiological tests, and they're certainly used in reference labs, places such as the CDC, and even DHEC nowadays uses a lot of molecular tests. This might be what a culture bench looks like. I think I took that picture probably 20 years ago. I mean, in the VA hospital here. I think it probably wouldn't look much different today. I haven't been in there recently. But basically, we have plates and we have simple strips that each with little cupules in them that tell us sort of what, say whether they're producing carbohydrates and what have you. For example, you might grow an organism on galactose. If it likes galactose, then it would metabolize it, produce an acid. The little cupule would change color because there'd be a diet indicator in there. So a colour change might indicate whether an organism is growing on a particular sugar, utilising a particular sugar. It might use an enzyme that might chew up a substrate, and that will produce a coloured product. The bottom line is simple metabolic tests and simple colour reactions that determine, and there are hundreds of these tests, and there's no way on God's green earth that we're going to go through every one of them. Some courses, they go through a lot of them. I think it's really overkill. But anyway, so we will cover examples of these tests, and that will be a case to really get a better idea of what they do. And we get into particular lectures, like in the strep, we will have a couple of examples in the Enterobacteriaceae lecture. We'll have some examples in the Streptococcus lecture. But the general point is this: they grow, they utilise, they produce, and somehow or other you end up with a colour change, and that tells you whether that particular material has been utilised or produced. Okay, so that's the physiological tests. And finally, antibiotic susceptibility testing. There are basically two ways you can do this. And I'll give you one example here, and we're going to get into another example in our growth metabolism lecture later. Okay, I'm a simple person, so I like simple slides. This is basically how you do antibiotic susceptibility testing on a plate. This shows you what you would do is you would streak the organism all over the plate, and you put a disc in the middle. The disc would be impregnated with a particular antibiotic that you were testing to see whether the organism was killed or not by it. If the organism basically you'll see that we see this grey colouring. What this is indicating, they haven't grown around the disc. In the case of this, the case where the organism couldn't care less about the antibiotic, it's not susceptible, the organism grows all over the plate, even right up to the disc. Because what happens is the antibiotic is impregnated on the disc, and when you put it on the agar plate, the antibiotic diffuses out of the disc into the plate. And basically what happens is there's a concentration gradient. The further away you get from the plate, the lower the concentration of the antibiotic. So the bottom line is out here somewhere there may be some antibiotic, but it's at so low a concentration that it doesn't do anything. So what this can tell you is not just whether the organism grows, but how susceptible it is by how close to the disc does the organism grow. That's antibiotic susceptibility testing on a plate. Okay, now, I can't stress this enough because I don't want anybody to go up to the Mayo Clinic somewhere, or worse than that, go into the VA hospital one day and say, could you give me the genomic sequence of this organism, please? And they'll look and say, what planet do you come from? Okay, please, okay, so it is important that we understand, how we develop tests and and what have you, but we do not do genomics at the moment in the routine laboratory. I would say that in some major places at the moment, they are actually certainly sequencing some of the genes from organisms, of any interesting organism, in some cases all the organisms they come across. But this is not done for routine diagnostics. It's not done for patient diagnostics. It's done more so that you can use this information in the future, basically, for other purposes. But if we want to really study organisms and get the maximum amount of information out of them, and also, with having the sequence of the entire genomes of organisms, and I think there's several hundred now, the entire genomes have been sequenced, it's going up logarithmically as the costs go down. Having this information, somebody can come along and say, aha, well, that's interesting. We've looked at this genome, and there seems to be a few genes that are present in this organism, and this is other organism that causes this nasty disease. Wouldn't it be nice if we could look at that gene and see if it really worked in a diagnostic kit? So that's where genomics comes in. It's been very helpful to the microbiology and infectious disease community, but it's certainly not something that we do in a clinical laboratory. Again, gene characterization. Sometimes it could be nowadays with some help from the genomics information, sometimes not. But you would take a particular gene that you're interested in. I've already mentioned the 16S RNA as being among the most common ones in our earlier lecture. You would sequence the gene. That will give you information. Or you can do what's called PCR, and I'll show you how that works in a little bit if you don't know already. The PCR is the polymerase chain reaction, and it is one of the things that's revolutionized certainly revolutionized microbiology, and has revolutionized some aspects of infectious diseases as well. The PCR is basically making a lot from a little. You would have a tiny, tiny quantity of bacterial DNA in a clinical sample, for example. So little you couldn't do anything with it. And then you go ahead and use PCR, and you make lots of it. And all of a sudden, you've got so much DNA you don't know what to do with it. And then you can start doing standard molecular biology tests on it. One of these standard tests would be restriction digests. One of the common sorts of techniques that's used in addition to the polymerase chain reaction is restriction digest. There are certain enzymes that will only recognize particular sequences or regions of DNA. And you can take a gene and cut it into pieces with restriction enzymes. And this will tell you it is used quite commonly to differentiate bacterial species. Hybridization is a technique that was used, and I'm going to show you an example of that, get into more detail in a second. Hybridization is something that's been used a lot in the past. It really has sort of gone away nowadays, although it is a very useful technique, and some diehards still use it. And this is history. Percent guanine plus cytosine. Nobody does that anymore. Basically, the idea was by looking at the percent GC or guanine cytosine, you could get some idea of the differences between different species. Well, it's so easy to see, it's so easy and quick cheap to sequence nowadays that nobody's going to use a percent guanine cytosine nowadays unless they're out of their minds. I'm going to explain to you what hybridization is, and I'm going to show you what PCR is a little more detail. I shouldn't have to explain to you what sequencing is, but it says by definition, sequencing, getting the DNA sequence. Okay, as an example of telling species apart, first at the level of the taxonomy level, then in a clinical kit. 16S RNA sequencing differentiates bacterial species. There are, as I said already, there are regions of the 16S RNA which are conserved across the whole kingdom because we need this molecule in protein biosynthesis. On the other hand, there are regions that differ between bacterial species. And it's just an example because we know nowadays that this is not the only gene that we can use. There are lots of other genes, and I just use it as the one that was used most commonly. So this is probably the most commonly one used. And you can develop clinical tests based on this sequence. Once you know that, for example, that there's a particular sequence in one species and a particular sequence in another, then basically you can take the polymerase chain reaction and you can amplify only that sequence. And if it shows up, well, it's that species. If it doesn't, if it shows up, it's one species. If it doesn't, it's the other. So again, the sequence, knowing the sequence allows you to devise diagnostic kits, and the polymerase chain reaction is one example, one of the most commonly used. So let's talk about the current variant of the polymerase chain reaction, since basically the old classical polymerase chain reaction has pretty well gone away, and this one's a hell of a lot easier to explain anyway. This is what you would see used when it's used in a clinical laboratory, and it is in places like DHEC, for example, would use this. The CDC would use this. Some of the bigger clinical laboratories would do this. Basically, you take some DNA that you've isolated from the bacteria, and there are simple kits that you can get it out in a matter of seconds or minutes. It's not a big deal with clinical samples, very quickly. And what you do is, you might have, in a bacterial infection, there will not be many bacteria in a body fluid. There might be, who knows, thousands. Bacterial cell weighs about 10 to the minus 15 of a gram. So 10 to the 3 bacteria is basically nothing. Can't detect nothing, or next to nothing. You have to have lots of it to analyze in most situations. What the polymerase chain reaction does is it amplifies particular genes. And what happens is there's a dye that's present which is sitting in the solution with the everything else. And this dye is not coloured. As the amount of double-stranded DNA is amplified by this enzymatic amplification reaction, and yes, there are details missing here, and yes, they're purposely missed out. But as the DNA amplifies, what happens is the non-coloured dye binds to the double-stranded DNA, and when it binds, it becomes fluorescent. And so in this thing, all you simply need to do is just throw your material into this machine with the right stuff, and just simply watch and say, hey, did it go green, or did it become a fluorescent green or not? That fits with the clinical microbiology though. It's sophisticated, but very simple to learn and do. And that's become one of the, if you look in the Journal of Clinical Microbiology, for example, probably not so much this year, but over the past few years, you'd see it. If you go back to the old issue, you see almost every article had the word PCR somewhere in the title. So that's a very, very common technique that's used now. It can be used for non-culture-based detection, or it can be used as a simple way to do a genetic test. But again, we're using it here not to study genomics or genetics or anything. It's a kit. You know what you're looking at. You know what gene you want to show or what variant of a gene. Is it there? Isn't it there? What have you. That's how you use this technique. As I mentioned already, DNA hybridization is a much technically less sophisticated technique. It gives very useful information. But pretty well nobody does it in this day and age. There are a few diehards, as I say. But it is a useful technique, and it's still, people expect you to know about it. So let me explain what DNA hybridization is. And again, it's not something you would generally do in a clinical microbiology laboratory, except for the fact that I'm going to use the word probes. And probes are something which are kits. And so the principle of how a probe works is the same principle of how DNA hybridization works. So I do want you to know what a probe is. So let's go through DNA hybridization first. Okay, we would isolate the DNA from one strain of a particular organism, and we'd isolate the DNA from another organism. The first DNA we would stick on a membrane. Actually, excuse me, first we would break the double-stranded DNA into single-stranded DNA by heat. Then we would stick it on a membrane. So there would now be single-stranded DNA stuck on a membrane. You take the second strain of DNA and you do the same thing, and you end up with single strands of the second strain. You take this solution of this DNA from the second strain and you mix it with the first strain. Now, we all know that the two strands of DNA recognize each other. If the DNA is very different, then basically the one strand of DNA will not recognize the other strand of DNA. If they're very similar, they will bind very strongly. So in the two worst case scenarios, what will happen if there's essentially no homology, none of the DNA from this strain would stick to the membrane. That would be 0% homology. In the other case, for the second strain, if it's essentially all of it stuck, that will be defined as 100% homology. And these are definitions. It doesn't mean anything beyond that. Just these are the two extremes you can get. Basically, none of it sticking, essentially very little or a lot sticking. And what you can do then is there are various ways which aren't worth going into. You can measure very simply the amount of DNA that binds or doesn't bind, and that gives you a number. And for example, two species were in the past considered a single species if they show 70% or greater DNA homology. You don't need to memorize that number. I'm just giving you it as an example. So that's how you would use the test. Again, it's not done in the clinical microbiology laboratory, and it's rarely done nowadays outside of a few labs anyway, because genomics is beginning to take over. Some other things. Now that was genetic tests that we talked about. Basically, is a particular gene there? Isn't it there? Is it slightly different in sequence between one organism and another? If we're developing these tests and working them out, that's taxonomy. If we're using them and we're going somewhere with this information in an applied real-world situation, that's the clinical microbiology laboratory. Okay? All right, now there's other things. This is actually something that's been used an awful lot. As the years have gone by, we go in sort of cycles. We're in another cycle at the moment. But basically, as the molecular biology revolution came along, which there were a number of Nobel Prizes awarded for, including for PCR, and basically these other things have sort of begun to disappear. But the most common biochemical test that's used is to look for the presence of particular long-chain fatty acids, particular chain lengths and degrees of saturation and what have you. Basically all you need to know is what fatty acids are present in a particular organism. That's used taxonomically, was used a lot. There are a couple of companies that you can send a sample to and they'll do it for you for $50-100 or something. Particularly used a lot was something that sounds the same, but it isn't. These are structural fatty acids present in the membranes I'm talking about here. An older test, which is still used, is looking at the short-chain products, short-chain alcohols and fatty acids. And these are metabolic products. It says fatty acids in both cases, but it's got nothing to do with each other. That's long-chain structural fatty acids. These are short-chain metabolic fatty acids and alcohols. We'll get more into this test when we talk about our metabolism lecture. Okay, so these are a couple of examples of things that are done at the chemical level to differentiate things. Again, most of the time, too complicated, wouldn't be used in the routine clinical laboratory. We're in another age at the moment, and now we've gone into the proteomic age, and there are a lot of laboratories beginning to do this because it's so simple, and so I think you will come across this. And basically, it consists of extract and inject. As I wrote in a recent invited review in the Journal of Clinical Microbiology on the subject, Basically what it comes down to is extract and inject. You take a bacterial sample, you extract, inject it in the machine, it gives you the molecular weights of proteins. It takes about as long as I said to do it. Unfortunately, it's a chemical technique, so a lot of people are a bit frightened of it at the moment. But I just want to mention it because it is becoming very common, and I'm sure will become even more common by the time you start going into practice. Okay, rapid diagnosis without culture. When and why? There are situations where you just have got no choice. For example, some cases like syphilis, you might think in this day and age, we know all we need to know about bacteria. I wish. For example, Treponema pallidum, the causative agent of syphilis, has been been known by mankind for hundreds of years to be a sexually transmitted disease. We still can't grow this organism in the laboratory. So there's no way, God's green earth, that you could use a culture-based technique for diagnosing syphilis. We'll get into that details later. Mycobacteria, tuberculosis, the causative agent of consumption or TB, it used to take about 6 weeks to grow up, it takes about 3 weeks still to grow the organism up. There's no way you can do rapid culture when it takes 3 weeks to grow an organism up in the laboratory. So there are alternatives. That's why we even bother to think about rapid diagnosis without culture. The oldest example, probably, of the non-culture based, and many of you have heard of this, and if you haven't heard of it, you've certainly been used on you, is the streptococcal agglutination test, or nowadays we call it the rapid strep test. And I'll put it, this version is not really used anymore, but it's the simplest way to explain it. So I've got it here for now. Basically, all you do is you take some latex beads, and you have attached to the latex beads, you would have an antibody. You take a throat swab from a patient, and it would have streptococci from streptococcal pharyngitis or sore throat. You simply mix the beads with the throat swab extract, and what happens is the antibody recognizes the antigen and forms a complex. And so all that happens is you just look at it and you say, well, are the beads the smooth white color, or are there particles present? It actually works very well. The problem is it can be a little hard to interpret sometimes. So newer versions are not necessarily much better in that sense. They're just easier to read. So this is pretty well gone by this version. But this was the original, the oldest non-culture based method. Particularly nowadays when we think about detection without culture, such as it's been very successfully used in tuberculosis, is the PCR reaction. I've mentioned that here. PCR has turned out to be a wonderful technique which is used in all sorts of ways. And take a wander down to the VA lab sometime, or whatever hospital you happen to be in, and look and see whether they're using culture plates or whether they're doing PCR. And the issue is that if PCR which is designed to detect organisms without culture, if it was working wonderfully in every case, then you have to ask yourself, why is the lab full of culture plates and not real-time PCR machines? The issue is that when it works, it's wonderful. It works great when the organism has been grown up as a test. In those cases where you're trying to do it from clinical samples, some diseases it's turned out to be wonderful. Some cases it hasn't. So, but it's very widely used. And finally, microscopy. I think this is the final one. Well, maybe not. Two things. Okay, microscopy and then serology. Microscopy. Microscopy is about the simplest way that you can do non-culture-based detection. And those who do infectious disease residencies or postdoctoral fellowships, I'm sure you'll spend a lot of time just taking spinal fluids and things like that and looking under the microscope and looking at spinal fluids and what have you. Microscopy is great. If you can see, if you've got a body fluid from a patient and you see some organisms there, you know that person's got a disease, and probably you know what the disease is. Unfortunately, it's tiring, and there's a limit to how long somebody can spend looking down a microscope. Usually speaking, you're only lucky about maybe 50% of the time. That's a very rough estimate. So if microscopy works, if you see something, you've got rapid diagnosis. If you don't see something, well, you're unlucky and you don't know what's going on. So microscopy is a useful technique, but not perfect. Not used very much, but you do see serology used. And I want to stress, I know we're at the end of the lecture here, but people get confused by this. So I want to give you a minute or two on this. Please don't get confused with antigens and antibodies. You have had 20-odd lectures now in immunology. You should know the difference, all right? When I talked about the agglutination test, we were looking at antigen. Okay? That was not serology. Serology refers to serum. In serology, you're looking at the levels of antibodies in a patient. In the case of the antigen, the rapid strep test kit, the antigen's there. You don't have to do anything to it. You extract it because it's there. In the case of serology, it takes weeks, sometimes many weeks, for a decent antibody response in a bacterial infection. So you would never, ever, ever use serology for rapid diagnosis because it takes a long time for the antibody response to develop. So in serology, we're looking at the antibody response. It's done after the event. If you had somebody and we'll get into my strep lecture as an example of why. If you're in a situation where you don't know if a particular organism caused a particular disease, you're in trouble. Well, if it's long gone, there's nothing you can do about it. You can come back afterwards and you say, well, hang on a minute, the organism's gone, the patient's no longer got an active infection, but I wonder if they had that infection. And then you can go back and see if there's particular antibodies against that organism floating around in the bloodstream of the patient. That's serology. Okay, so don't confuse serology with detecting an antigen, basically an antigenemia or antigens. Okay? Okay, I think that's it. I'll be happy if anybody's got any questions to stick around. Thank you very much.